Hey, Dr. Judd.
Hello. Let's see.
Did you start recording?
I think it automatically did. I could, I
Okay.
think I'll just post it. I can take care of that. Post it. Here it is.
I'm having trouble hearing.
Testing. Talking for you to
Oh,
check.
I got it. I've
Good.
got it. I got it now. How was everyone's Thanksgiving? Did you guys eat? Thumbs up from Alexis.
Way too much.
Is in trouble. Who ate too much?
Keaton.
Oh, Keaton.
Nice. Okay, close that. Modal.
You guys have all seen the inside of Dr. Judd's house, but now you're seeing our upstairs office. How many of you are on
Nice.
campus right now?
1, 2, 3.
What about the rest
4.
of you? Okay, so quite a few of you are still on campus. Okay.
Yeah.
How are you getting food while you're on campus? Is the tap open? Okay. All right.
That's good. Okay.
Okay. Tomorrow in lab for my group, I'll meet you at 7:45 on Zoom. We're going to talk about your presentations and we're going to talk about your data somewhat. Dr. Judd and I are still working through some of the data. We had troubles with some of the data in terms of completeness and things like that over the holiday. So my plan is going to be to post screencasts that walk you through the data that you can go and look at, as well as we'll have the scripts and the data files posted, especially for those data files where the Tuesday and Thursday labs were both working on a particular data set.
Yeah.
So I'm having headphone difficulties. All right. Dr. Judd, do you have any announcements?
Just, I was going to say similarly that we're going to go through the scripts together over Zoom, but you guys will also have the scripts so you can follow along. You can go over it again afterwards. And we're just trying to get plenty of comments on those and make sure that it's clear when data are coming from one lab or the other, because there's a lot of overlap, which is good, but there's also places where there's not so much overlap between the two lab sections. So we'll be dealing with that.
Yeah, for example, the pollen limitation groups that were looking at stigmas, it seems that one group measured floret length and the other group measured inflorescence length, I think. So there are some differences that we have to account for. So there's that.
I was also going to remind everyone that the VNet all reviews are due this afternoon. I think we've got it posted as 5 for the due date, 5 p.m. Yeah.
These are your comments on other people's papers, and I started going through your VNet et al. Reviews for the faculty portion, And I'm still working on those as I get them done. I'll be sending them out to you so that you have feedback. Be sure that you are removing yourself far enough away from the original text that you're not risking plagiarism. So we've given you some tools for doing that. Make sure that you're not plagiarizing. I think we're going to have 2 more honor code violations from the VNet et al. Papers. Oh, sorry, no, yeah, VNet et al. Papers. So just be aware of that. Write in a way that promotes understanding, and write in a way that promotes understanding while at the same time avoiding the original language of the original paper. If you have any troubles with understanding the Brody paper, let us know, and we'll do our best to walk you through that stuff as we go. Some of what we're going to talk about today might actually help with that somewhat today and on Wednesday. Today we're going to be revisiting concepts in quantitative genetics and expanding on quantitative genetics beyond what we covered during the Galapagos case study. And then on Wednesday, Dr. Judd is going to be revisiting the idea of an adaptive landscape, but in the context of multivariate evolution rather than single traits. The PowerPoint that is up on the Moodle site actually takes you through both of those lectures, but I'm not sure how much of the second part of this presentation Dr. Judd was planning on using or not. He might have some changes. So when I get to the end of today's material, you'll see another set of learning objectives and then a bunch of other slides that we won't cover today. And those are what we've used in the past for adaptive landscapes. So, all right. Let's get started. Can you guys see my screen? No, I'm getting shakes of the head no. How about now? All right, cool. So we've already talked some about quantitative genetics. So we're going to be revisiting some material that we did in the past. And we're going to be, like I said, expanding to talking about more than one trait. So you should be able to describe what quantitative traits are. We've already calculated the evolutionary response to selection for a single trait in the Galapagos Islands case study. We'll ask you to do that again. We'll also have you now looking at multiple traits so that we can look at genetic correlations between traits. So you should be able to recognize where positive and negative genetic correlations exist. And using this information, we'll show you how to calculate the evolutionary response to selection for genetically correlated traits. And it involves a little bit of matrix algebra, but it's not matrix algebra that's so complicated that you can't grasp it. And then the goal of all of this is so that you have an understanding of how genetic correlations between traits affects the evolutionary response to selection. And so these are, if you can do these things based on today's material, you'll be well set for what comes ahead. So we use quantitative genetics to study quantitative traits. Who can remind me what a quantitative trait is?
It has to do with numerical value when describing a subject, right?
Yeah, so go into that a little bit more. Who was that?
Daniel.
Daniel, yeah. Go a little more into that.
So like the graphs that we had to analyze about the grazed and ungrazed, those were all quantitative values.
Okay, so those are quantitative values. In that case, though, those are community sorts of values. So those aren't the phenotypes of individuals. What would be a quantitative trait in you?
I have 2 legs and 2 arms.
You do have 2 legs and 2 arms, so that would certainly be a quantitative trait. Unfortunately, everybody has 2 legs and 2 arms, and so there's no variance for those. What would be a variable trait?
Could speed be variable?
Yeah, speed could definitely be variable. Who was that?
Riley.
Oh, Riley, okay. Yeah, speed would definitely be a trait that would be under the control of multiple genes all contributing to that trait. And then there's a lot of variation between one individual and another in terms of how fast they are. You have Usain Bolt, and then you have the rest of us, for example. So these quantitative traits are traits that are underlain by multiple genes that all contribute to that single trait. And in most cases, one of the alleles contributes a little bit to that trait while the other allele does not contribute to that trait. And so height, milk production in cows, weight of cows, number of eggs that chickens lay, et cetera, all of these kinds of traits would be considered quantitative traits. The math, ultimately, we've talked about how we deal with the math of these Mendelian, these quantitative traits. The math grows out of the Mendelian inheritance of single genes, but because we're looking at multiple genes, these things take on distributions rather than, yellow seed pods versus green seed pods sorts of traits. And the goal of quantitative genetics is, of course, to predict how selection acting on one generation will produce genetic change in the next. So these are polygenic traits. Each dominant allele affects the gene. Each dominant allele adds to the effect of the genes on the phenotype. And once again, these are some more examples. I forgot about ear length in corn. Crop yields. I think a lot about agriculture because I teach a course that deals with agriculture a lot. So all of these kinds of traits. So this is all review. Imagine that you have 3 genes. You have these 2 heterozygotes. You're heterozygous for gene A, gene B, and gene C. You cross those individuals, and we get these genotypes. And once again, this has the same kind of symmetry that we've seen in other Punnett squares, where you have the homozygous dominant for all 3 genes up here, the homozygous recessive for all 3 genes down here. Along the diagonal, you see that everybody is heterozygous for all 3 traits. And as you move from the upper left to the lower right, you get fewer and fewer dominant alleles. If we just count up the number of dominant alleles, it looks like this. And once again, you can see the symmetry. Along the top, you have 6, 5, 5, 4, 5, 4, 4, 3 dominant alleles. Along the first column here, you have 6, 5, 5, 4, 5, 4, 4, 3. And so once again, this symmetry in the Punnett square is nice and regular. It's nice when things work that way. And it gives you this normal distribution if you look at the number of contributing genes or contributing alleles to the phenotype. Once again, this very symmetrical normal distribution. And we talked to you before about how you can measure heritability, and we'll go through that again. And if you know what the heritability of a trait is, and you know how much selection is acting on that trait, you can predict how that trait will respond evolutionarily in the future. So the evolutionary response to selection is equal to the heritability multiplied times the strength of selection. In the same way, if you don't feel like measuring heritability, you could always do something that exerts selection on a population, observe the evolutionary response of that selection, and then use the evolutionary response and selection to then calculate what heritability is. And there are some biologists who actually measure heritability of traits this way. So this equation is easily manipulable to basically, if you know 2 of the things, you can figure out what the third thing is. So this becomes a more complicated matter when we start thinking about multiple traits. So Little Butch Brody, for his doctoral dissertation, worked on this species called Thamnophis ordinoides. It's a slug and worm eating snake in the Pacific Northwest. And these 2 species come in 2 kind of distinctive forms. There's a striped form and there is a more checkered form. And there is there's continuous variation between stripedness and checkered, but these would be the two ends of the extreme, a very striped snake and a very checkered snake. And when he was running these snakes down racetracks to test their sprint speed, he noticed that some individuals tended to just go straight down the track without any difficulties, and other individuals would go partway down the track and then they would turn around on themselves and go the other direction. Or in some cases, they would turn around and stop and not continue down the racetrack. And so he started scoring their tendency to reverse themselves as well. And what he discovered was that stripedness tended to occur in snakes that tended to run in a straight line, and reversals tended to occur in snakes that are checkered. And so he found that these two phenotypic characters tended to be related to one another. Well, you can calculate heritability by looking at the stripedness of the parent and stripedness of the offspring. And when you do this, you find that the heritability is 0.707, because once again, the heritability is the slope value of this relationship. If selection acts on stripedness, this is similar to a figure that we've given you before, then what you can expect is a certain response to selection. And remember that you can designate the response to selection with a capital R, or you can also designate it as a delta Z, which is change in Z, where Z is the phenotypic value. So you can just as easily have put R here. You'll see authors using both notations. So because this trait is heritable, when you exert selection on the parents, you get an evolutionary response to selection in the offspring, because some of these not very striped snakes are not reproducing, whereas all of these striped snakes are reproducing. If you know what the value is, if you know what the change in the value is here, you can calculate the difference in selection from a value of 1542 after selection occurred compared to a value of 13.13 before selection. So this difference is a positive difference of 2.29, and that's the direction of selection. So this is a directional selection scenario. The heritability is 0.707, and so you should expect the response to selection to be an increase in stripedness of about 1.62, and that seems like pretty close to what you would expect there. You can also solve this a number of different ways. You could actually just plug it into the equation, where you basically look at the slope, the post-selection mean of the parents, and the y-intercept, which gives you what you expect the post-selection mean of the offspring to be. And then you can subtract that post-selection mean, or subtract the pre-selection mean rather from the post-selection mean, and it should give you the same answer. And so this heritability equation basically relates selection to the change in phenotype. And so you can also use that equation this way. I find it easier to think of the breeder's equation, this equation at the top, but that's not the only way that you can think your way through this whole process. You can do the same thing with reversals. If reversals are heritable, are reversals more or less heritable than stripedness was? Here's stripedness. Here's reversals. Which one has higher heritability?
Reversal is higher.
So here's stripedness. And here's reversals. What do you look at to see what the heritability is, Riley?
Just kidding. It's the slope. It would be the other one.
Yeah. So reversals has a lower slope of 0.245, whereas stripedness has a slope of 0.707. Good. Good. All right, so the same thing applies here. If you have selection, in this case, selection is going the opposite direction such that individuals are becoming less prone to reverse themselves, then you would also expect there to be a response to selection. In this case, the selection is moving in the negative direction, and so the evolutionary response to selection is also in the negative direction. And once again, we could figure out what the strength of selection was, multiply that times the slope, and we could look at the evolutionary response to selection, and it's a decrease in this case of selection of 0.14. We could also figure it out using the equation of the line and then comparing the average offspring post-selection to the average offspring pre-selection, and we should get the same result in both of those cases. Once again, just relatively simple math. So we're talking about directional selection in this case, where we have the population moving one direction or the other direction. Don't forget that there's also stabilizing selection and disruptive selection in all of this. Remember that disruptive selection is what we studied in the Galapagos finches at the beginning of the semester. But once again, it's always effects that selection is having on the position and shape of this quantitative variation that we see in populations. So we can measure selection on multiple traits simultaneously. And so this is one of the things that Butch Brodie did. He went out into the field and he got a bunch of gravid female snakes. And remember when we went through the introduction to the case study, I showed you a picture of little Butch Brodie holding up some snakes, and you could see the lumps in the belly of the snakes, which were actually embryos that were getting ready to be born. And when the females gave birth to their offspring, because this particular group of snakes gives live birth, they would score the females for stripedness and reversals, and they would also score all of the offspring. So they had a parent value for stripedness and reversals, and they had offspring values for stripedness and reversals. And this is Big Butch, Edmund Brodie Jr., racing one of these snakes down a racetrack. I have a similar but smaller racetrack down in my lab. And so they measured speed, they measured stripedness, they measured reversals. But the paper that we're going to be thinking about just looked at stripedness and reversals primarily. They invented a way of quantifying stripedness, so they would score various aspects of the color pattern, and so that they could arrive at a numerical assessment of what stripedness is. And the reversals was just, how many times they reversed over the course of running them around a circular racetrack for a certain distance. So when you think about these, yeah, go ahead.
Did they take average number of reversals across multiple runs?
Oh, I don't recall. It seems like the type of thing that they would do.
I think that's right, because their values are continuous. There's decimal values in the reversal.
Yeah, probably. Yeah, it wouldn't surprise me. So they had two types of racetracks. They had one type of racetrack that was a linear racetrack for measuring speed, and then they had a circular racetrack that you crawled under and then popped up in the inside of,
Gotcha.
And then you would chase them around in a circle so that they could run around in a circle, and then they would reverse themselves, and then they'd go the other direction, and they would reverse themselves again, go back the first direction, and back and forth, pretty much until they kind of exhausted themselves.
Yeah.
And my guess is they probably did this multiple times.
Yeah.
They did all of this at UT Arlington when I was a student there, and they spent a lot of time down in the snake chasing room. When you have two traits, these traits can be genetically coupled in the same way that your arm length and leg length are genetically correlated with one another, mainly because they're under the control of the same hormones, growth factors. And as a result, when you measure your arm length and your leg length, they are similar. They're correlated with one another. You don't tend to have people with really long legs with really short arms. If you're long-legged, you're also long-armed. And so what happens is when selection acts on one trait, it can affect the other trait evolutionarily because of these genetic correlations. And we can actually measure these genetic correlations. And one way of measuring the genetic correlation is similar to the way we measure heritability. And so if we want to look at the genetic correlation, we can ask, well, what's the correlation between stripedness and reversals? Well, if we measure reversals in the parent and stripedness in the offspring, we will get a relationship. And in this case, this would be a positive genetic correlation, such that females that reverse themselves a lot give rise to offspring that are very striped. On the other hand, you can have a negative genetic correlation where females that reverse themselves a lot give rise to offspring that do not have a lot of stripedness. They're more checkered in their color pattern. And in the case of this case study, the genetic correlation that they found between these snakes was this, such that females that reverse themselves a lot tended to be checkered. Females that tended to slither in a straight line and not turn around on themselves tended to be more striped. And there's good reasons for this. When you see a striped snake running, or a striped snake slithering in a straight line, there are biases in your vision that make that snake look longer than it actually is. And so if you try and grab that snake by the tail, oftentimes you'll grab air because your eye is continuing that stripe out beyond the end of the tail of the snake. And so it's an optical illusion. And so evolution has favored snakes in a way that when you're striped, it's harder to see where the snake begins and ends. And as a result, if you just keep on hauling ass through the grass, you tend to not get caught as often because of this optical illusion. Checkered snakes, when they go straight, that checkeredness just kind of all blurs together and they look like this one solid uniform snake. And it's relatively easy to make out that snake, but as soon as that snake stops, the vertebrate eye continues following where that snake was going to be going. And then when you realize that that snake isn't there anymore, you come back to where the snake was, that snake is stopped. And because it's stopped now, it looks checkered. And so it has this disruptive color background that then makes it harder to see when it's sitting there still. So these are two different modes of avoiding predation that have the stripe mode does one thing, the checkered mode does another thing, but both modes might be equally valuable at avoiding predation. What is not good is a striped snake that stops, because as soon as a striped snake stops, you lose that optical, I just lost the word.
Illusion.
I just said it. Illusion, the optical illusion, because the striped snake is bold when it's stopped. And the checkered snake, if it just keeps running in a straight line, it just looks like a snake slithering through the grass. And so to not have these two traits, a behavioral trait and a morphological trait, to have them uncoupled, where you have striped snakes that stop and reverse themselves, or checkered snakes that run in a straight line, that then destroys the anti-predator advantage of having that particular color pattern, because you have a mismatched behavior. And so this is what Brody was interested in exploring. So you look at this through the same equation, except now we have to make this equation multivariate. And so before, the heritability of the trait was multiplied times the selection to give you the response to selection. In this case, the response to selection is due to a genetic variance covariance matrix, which is what G is, and a selection vector, which is what beta is. Now I realize I've just said genetic variance covariance matrix, and I've just said a selection vector, and you're like, what on earth is he talking about? Well, I'm talking about this. So just to not frighten you, think of trait 1 and trait 2. The genetic variance is basically the correlation between trait 1 and trait 1. Trait 1 in parents and trait 1 in offspring. G12 is the correlation between parents and offspring of trait 1 in parents, for example, and trait 2 in offspring. G21 is trait 2 in parents and trait 1 in offspring. These two things are exactly the same value, and then G22 is the genetic correlation between trait 2 in parents and trait 2 in offspring. So G11 and G22 are basically like heritability. And these two elements, G12 and G21, is the genetic covariance between the two traits. Beta 1 is selection acting directly on trait 1, and beta 2 is selection that acts on trait 2. And so selection acting on trait 1 can have an effect on trait 1, but selection acting on trait 2 can also have an effect on trait 1 due to this genetic correlation, if that genetic correlation is non-zero. The way you figure this out is you multiply G11 times beta 1, and you add that to G12 times beta 2. That gives you the response for trait 1. For the response to trait 2, you take G21, multiply it times beta 1, and G22, and multiply it times beta 2. You can also think of it by row and column if you want. It doesn't really matter how you skin this particular cat. This is just one other way of thinking about it. If we want to think about the actual case of the snakes, we could actually convert this to reversals and stripedness, where GRR is the genetic variance or heritability for reversals, GSS is heritability for stripedness, and GRS and GSR is the correlation between those two traits, the genetic correlation between those two traits. And we don't generally call these correlations any longer. We call them covariances and variances, but it's really, there are some fine scale reasons why we do that because of the way we calculate these things, because we want to actually look at the influence of the genes underlying reversals as independent from the genes underlying stripedness. And so the methods that we use to estimate these are somewhat different. But you can think of them essentially as heritabilities and genetic correlations. So we can measure the relationship between offspring and parents with reversals. We could look at this for stripedness, and we could also look at stripedness versus reversals. And this would now then tell us what the genetic covariance is between these two things. And once again, females that reverse themselves a lot tend to be checkered. Females that do not reverse themselves a lot give rise to striped offspring. And so we would generally expect genetic variances to be positive, and once again, covariances could be either positive or negative, or zero for that matter. In this case, they just happen to be negative. So selection is then a vector where we have the direct selection on trait 1 and the direct selection on trait 2. And this is just measured in terms of, in the case of reversals, how much does the value of the parents change before selection to after selection. So it's all the same set of ideas that we had before. In this case, it's just dealing with multiple traits. Same thing with stripedness. And then the response to selection is just what happens through both direct selection acting on heritability and indirect selection acting on the genetic correlation or covariance. So this is what it looks like. If we have the genetic variance covariance matrix and the vector of selection, G11 times beta 1 plus G12 times beta 2, that's the response for trait 1. G21 times beta 1 plus G22 times beta 2 is a response for trait 2.
For those of you who haven't seen matrix algebra before, the order in which these things are multiplied and added together and the way they're color-coded is a standard way that we multiply matrices and vectors. So the beta column there, that's a vector. G11, G12, G21, and G22 is a matrix, and you always go in the order shown here and depicted on a previous slide.
One way of thinking about it is to take the top row here and turn it into a vector where G11 is here and G12 is here, and then you multiply across is another way of thinking about it. Then you would take this row here, once again, turn it on its on its into a vector where G21 is here, G22 is here, and then you multiply it across. Once again, whatever way you have to think about it, it doesn't really matter, as long as you can reproduce the math. If you go to graduate school someday and you're in like an ecology program, they might actually require you to go and take a course in matrix algebra, and you will spend a miserable semester learning matrix algebra, simply so that you can use a set of techniques for projecting population growth in stage structure populations, which are common to plants. So if you are an animal biologist, you will come to resent your matrix algebra class. If you're a plant biologist, you will think it's the greatest thing since sliced bread. Dr. Judd, do you have any comments about that? Did you have to take a matrix algebra class when you were in grad school?
You know, I didn't. Not in my program because I was mostly at the museum. But
yeah, okay.
I have had to brute force learn it a few times. So
yeah. But do you, in ecology class, do you take them through stage structure population growth models?
No, but we did go through. I decided not to do that this time. Last time we did that, and it didn't go well, and I wanted to take a different approach. But we did end up doing some matrix algebra, so you will see it again. Okay.
Yeah, there's a particular way of projecting population growth that relies heavily on matrix algebra, and it can get quite naughty, quite complicated. You can do this for as many traits as you want, as long as you can measure the heritability and the genetic covariance between the traits. Once again, in this case, we're going to stick to 2 just to keep it simple, and also 2 was mainly what they did. So you can think of this, once again, conceptually as G1 times beta 1 is the change in the mean of trait 1 due to selection acting directly on trait 2, and G12 times beta 2 is the change in the mean of trait 1 due to selection acting directly on trait 2. So this is an indirect form of selection because selection on trait 2 is affecting the response of trait 1 mediated by this genetic correlation. So here we have the matrix written out for reversals and stripes, and we can basically just rephrase all of that we just phrased earlier to talk about stripedness and reversal. So this is what response for reversals would be. It's the heritability reversals times selection acting directly on reversals times the genetic correlation between reversal and stripe times the selection acting on stripe. Similar analogous thing going on down here. So you're probably looking at me going like, okay, how on earth does this actually work? Well, presumably you've taken the time to measure what selection is on these 2 traits, and you've taken the time to measure both the genetic variance of reversals and stripe and the genetic covariance between reversals and stripe. You can do this just by having parents and offspring that you scored for all of these things, and it's just a matter of which set of correlations you're looking at. How would you measure selection on reversals and stripe? You have all these babies in the lab, you've scored them for reversals, you've scored them for stripedness. How are you going to determine how selection operates? Anybody? When you get selected against in the kind of purest, most harsh sense, what's the difference between someone who has been selected against versus not?
one individual lives and one individual dies?
Yeah, one individual lives, one individual dies. So somebody else other than Joseph, what could you do with these snakes? Well, let me give you a little more natural history about them. Butch Brodie would go and collect these snakes at hibernacula in the spring. Hibernacula are underground cavities where all these snakes come and overwinter. And then in the spring, they emerge from these hibernacula and they sun themselves in the early spring. And then when it gets cold at night, they go back into the hibernaculum and they come back out and they sun themselves. And then they do that over and over again until it finally gets warm enough throughout the night that they finally disperse away from the hibernacula. And then at the end of the year, as winter is coming on, they migrate back to the hibernaculum and then go underground again and wait out the winter there until they emerge again in the spring. And so he goes there in the spring, collects these snakes, because when they're coming out of the hibernaculum in springtime is when the females mate with the males. And so he knows that those females have been mated and they will eventually produce offspring. So now you have the babies. What could you do with the babies?
You could see how many of each type of pattern made it back to the like hibernating space each time.
Yeah, exactly. You could, after you've measured them for reversals and stripiness, you turn them loose into the field. You know what the phenotypes are of everybody that you turned out. You've marked them, and there are ways of marking snakes so that you know who's who when they come back. And when they come back, you can just look at who survived, who came back to the hibernaculum at the end of the year. Now, in many cases, those individuals that didn't come back probably were killed and eaten by predators. But in some cases, those individuals that don't come back might have like gone over the mountain and they're in another watershed and they hibernated over in another place. But the assumption is that most of those individuals that don't come back because they aren't alive to come back. Even though that's not a hard and fast assumption that you can make about everybody, it's a reasonable assumption. At the very least, individuals leaving the study area and not coming back to the hibernaculum is actually just going to reduce the measure of selection that you have, unless stripedness or reversal somehow affects one's dispersal ability to disperse away from where they came from. And that's precisely what he did. He set these babies loose and then went to the hibernaculum in the fall when the snakes are coming back, and then basically just looked at who returned and scored them once again on the basis of reversals and stripedness to make sure.
This is a good exercise for, if I can jump in here, for thinking about your data and sources of error. When you're looking for a signal, like the selection for these traits, you want to think about what kinds of errors you might expect to be dealing with in your data set, like the difference between snakes dying versus leaving. And sometimes you'll find that, well, as long as the signal that I'm looking for is really strong, stronger than the effect of any error, then I'm in good shape. I have the power to detect a strong signal, which is what you're looking for, a strong effect. Sometimes you can find that your errors might bias in the opposite direction of what you're looking for. And as long as you're able to think through what the errors might mean and then what that means for your ability to detect an effect, you'll be able to do some good critical thinking about what could come next, or what the odds are that you're going to detect a signal that you expect to find.
Yeah, the assumption that Brodie is making in this case is a striped snake is just as likely to leave the study area as a checkered snake is. And a straight slithering snake is just as likely to leave as a snake that reverses itself a lot. Which of those two assumptions is the weaker assumption?
I'm sorry, can you repeat that? It kind of cut out for me.
Yeah, sure. So the assumption that he's making is that striped snakes are just as likely to leave the study site as checkered snakes are. And he's also assuming that snakes that tend to slither in a straight line are just as likely to leave the study areas as individuals who reverse themselves often are to leave the study area. Which of those two assumptions is the stronger assumption versus the weaker assumption?
Isn't the first one a stronger assumption that they're
Yeah,
all going to leave?
Yeah, it's a much stronger assumption to assume that color pattern doesn't have anything to do with dispersal ability, but the assumption that different modes of locomotion, different behaviors related to locomotion, that's a much weaker assumption, because if an animal is always slithering in a straight line, it will tend to cover more area, more distance over a given amount of time than an individual that's constantly reversing himself. So you can do what Dr. Judd just said and say, okay, well, which of these two assumptions is stronger versus weaker? And so you can think, okay, well, if some individuals don't come back, it might be due more to locomotion than it is to color pattern, for example. So we can think of any given snake existing on this plane of reversals and stripedness, from fewer reversals to many reversals and less stripedness to more stripedness. These are stripy snakes at the top. These are checkered snakes at the bottom. And we can think of individual snakes on this landscape, but we can also think of the population average on this graph as well. And if we start with the population average that is here, we can basically ask ourselves, where on this landscape of phenotypes is this population going to move based on what's happening to it in terms of selection and what's happening to it in terms of heritability and genetic correlations. So if we start off with really low selection on reversals and higher selection on stripedness, and we know that the heritability of reversals is 0.245 and the heritability of stripedness is 0.707, and we know that there's this value of negative correlation between them, we can just use matrix algebra to figure out what the response to selection is. It's 245 times 0.008 and negative 0.751 times 0.102. Notice here that selection is stronger on stripedness than it is on reversals. As a result of this, we get a response to selection, which is reversals going down slightly and stripedness going up slightly. The interesting thing that you should note is that that reversals are decreasing. The tendency to reverse oneself is decreasing in spite of the fact that there is directional selection to increase reversals. But because the selection on reversals is lower and the genetic correlation is strong, it leads to reversals changing evolutionarily in just the opposite direction of what you would expect reversals to be doing based on just a knowledge of selection alone. And this is why we use multivariate quantitative genetics, because knowing something about selection would make you think that the population is changing in one way, but because of this genetic correlation, the evolutionary response of that trait is actually going in the opposite direction. Interesting, given the conversation that Dr. Judd and I just had with you, that selection on stripedness is stronger than selection on reversals. And so it may be that the low level of selection on reversals here is actually due to some individuals leaving the population because of their locomotor ability, not having anything to do with actual loss of individuals due to their locomotor mode. What this looks like is we can look at the direct effect of selection acting on reversals. That changes reversals in one direction. So remember, reversals will be going up and down, moving left to right. And then we can look at the degree of change in phenotype due to selection acting directly on stripe. And so we might expect to see the population up here where you had a slight increase in reversals and a big increase in stripedness. And that's due to the combination of stronger heritability and stronger selection compared to weak selection and lower heritability for reversals. So we see reversals changing less than stripedness. But of course, this isn't the only thing that is going on. What also is going on is the indirect effects of selection on one trait affecting the other trait. For reversals, this looks like strong selection on stripedness acting on the genetic correlation, which is also strong, moving reversals back, and also the weak selection on reversals combined with the strong genetic correlation, which changes reversals. Sorry, that was stripedness. Strong selection of stripedness acting on the genetic correlation, and weak selection of reversals acting on the genetic correlation, which changes reversals not very much at all. And so what we get is, sorry, stripedness is affecting reversals here, and reversals are affecting stripedness here. So reversals are going down a lot because of strong selection due to stripe. Stripedness is changing hardly at all because of weak selection on reversals. That's what I'm trying to say. Is that clear, Nathan?
Yeah, I think that works that time. Yep.
Yeah, okay. So you end up with a decline in reversals, but an increase in stripedness, which is exactly what we showed. And these things aren't drawn to scale because there aren't any numbers on any of these axes. But you can see why when you have strong selection on stripedness, it can drag reversals quite far away from where they would have been without that genetic correlation, because selection on stripedness is strong, dragging reversals in exactly the opposite direction in which selection wants to push reversals. Same selection, but in this case, what has changed in this scenario? Has heritability changed? Heritability is 2.245 and 0.707. So heritability hasn't changed. What's changed? This scenario versus this scenario.
What's different about that G matrix?
Yeah, the selection gradients are the same. The G matrix has changed. How has it changed?
It's 0.184 instead of 0.751.
Yeah, so the genetic correlation is much less strong. So you might expect the effect of indirect selection, this correlated selection, acting on the two traits is actually less than it would be. And when we run through the math, what we find is that reversals are still going in the opposite direction, and stripes are still going in the direction that they were going before, but the effects are not nearly as strong. When we look at that in this kind of scenario, what we see is the effects of direct selection are still there, but when we look at the effect of stripedness on reversals, we see that there's a much lesser effect because stripedness, it still has the same degree of selection, but the genetic correlation is weaker, which drags reversals less far back. And we see a similar thing happening. This weak selection acting on a weaker genetic correlation leads to less change in stripedness as well. And so we still don't end up exactly where we were when we would have ended up had we just had directional selection acting on the two traits independently, but it's not as far away as when that genetic correlation was really strong. And so strength of selection is important, but also the strength of the genetic correlation is important. What's changed in this case? The G matrix is what? Is it the second scenario we looked at or the first scenario we looked at? Connor, you remember the numbers.
Yeah, it's the first, I believe.
Yeah, this is the first scenario. So what's changed here? Somebody other than Connor.
The selection.
Selection on reversals versus stripe. Go ahead.
Oh, I know the second matrix changed.
Yeah, so this has changed such that there's higher selection acting on stripedness, right? So because there's higher selection on stripedness, acting on the negative genetic correlation between stripedness and reversals is going to affect reversals much more than it's going to affect stripedness, because selection on reversals is the same as it was before. So once again, when we work through the math, we see that reversals is going in a much greater negative direction than it was before, but that's not due to selection on reversals changing at all. It's due to selection on stripedness changing. Oh, that animation didn't come up right. Once again, we see selection on stripedness leads to a much bigger change in stripedness, but we have the same degree of change in reversals. But this is the effects of direct selection acting on those two things. And now the selection on stripedness drags reversals way back here, but the selection on reversals doesn't drag stripedness down very much at all because it's the same as it was in that first scenario. And so now we see a big change in reversals relative to what you would have expected if you didn't have this genetic correlation, and the similar change in stripedness as we had in scenario one. Once again, illustrating now the importance of selection on similar levels of genetic correlation. Last scenario, what's different about these? What's the primary difference?
The genetic correlation.
The genetic correlation, Katie, it's in this case what?
Now it's 0.651.
Yeah, so it's positive and it's big. It's big and positive. And so the selection is back to the selection that we saw in scenario one, condition one. The heritabilities are the same, but it's the genetic covariances that have changed. Now they're positive and they're large. This has the effect of increasing both reversals and stripedness in a positive direction. So that negative direction for reversals is gone. So in this case, we might expect stripedness to increase by a certain degree and reversals to increase by a certain degree. This is the same as the other ones, just drawn instead of drawing reversals here and stripe up there. They're just flipped for whatever reason. But then when you get to this point, you haven't considered that positive correlation. And so as reversals act on stripe, it increases stripe even more. And as stripe acts on reversals, it increases reversal even more. So that in this case, evolution is actually proceeding faster than you would expect if you were treating these two traits as independent of one another because of these genetic correlations. And so that's the change, because once again, the genetic correlations are positive, pushing both of these traits to get bigger and bigger. So when you think about the magnitude of the negative genetic correlation, how does the response to selection of reversals and stripedness operate? If you increase the size of the genetic correlation, what happens? or if you decrease it for that matter. If the genetic correlation is strong and negative, what do you expect to happen? Here you have a situation where there's a strong negative genetic correlation, positive selection for reversals, positive selection for stripedness, but what's the result?
Is it that it has the stronger heritability?
No, the heritabilities are the same in all of these. I'm looking for what's the effect of the negative genetic correlation. Positive selection on reversals, positive selection on stripes. Snakes should be getting more and more reversey and more striped, right? If you just look at selection. But what actually happens? It's down here.
Reversal is negative.
Yeah, the reversal is negative. That was Keaton.
Yes.
Yeah, the reversal is negative, and the rate of change of stripes is also positive, but it's not moving very fast. It's not changing very fast. And so in this case, those negative genetic correlations, when they're strong, they can either reverse the evolutionary response to selection compared to what you would expect selection to actually be doing, or it can slow things down so that the selection isn't having as big of an effect as it would have normally because of correlated selection. Yeah, I have to go cycle through all of these. Don't get carried away and overrun myself. How does the direction of the genetic correlation affect the evolutionary response to selection of reversals and stripedness? If it's negative, what does it do? We just covered that.
If it's negative, it'll be negative. If it's positive, it'll be positive.
Yeah, so the response to selection, if it's negative, it will tend to push things in negative directions. And if it's positive, it will tend to push trait change in a positive direction. How does the strength of selection affect the evolutionary response to selection of reversals and stripedness? Somebody other than Keaton. Keaton seems to be doing well with this. Cameron, give it a shot.
I'm not too sure.
What happens if we change the value of selection? Selection increases, what happens to the answer that you get in the end?
That'll increase as well.
Yeah, so selection just amplifies whatever is happening in the G matrix. So if selection is increasing here, then it acts both on the heritability and whatever the genetic covariance is. So you would just expect bigger changes in whatever it is as it increases in magnitude. So genetic correlations can either cancel out or slow down evolution when the genetic correlations are negative. Positive correlations can speed up evolution. But then the next question is kind of how do these negative genetic correlations come into being? And when we look at the adaptive landscape here, we see that when we look at the fitness of individuals, we find that striped snakes that don't reverse themselves very much have the highest fitness, and checkered snakes that reverse themselves a lot have the second highest fitness. Checkered snakes that reverse themselves a lot, checkered snakes that go in a straight line have low fitness, and striped snakes that reverse themselves a lot tend to have the lowest fitness. So this is this kind of a fitness landscape where there are two fitness peaks. One fitness peak is striped snakes that go in a straight line. There's another fitness peak of checkered snakes that reverse themselves. But striped snakes that reverse themselves a lot don't do well, and checkered snakes that run in a straight line don't do very well. And so we can think about this three-dimensional graph being flattened to create this topographic map with contours to show you where fitness peaks are and fitness valleys are. And as it turns out, this thing will then cause, in one case, the behavioral phenotype to become correlated with the morphological phenotype. And this is actually what Dr. Judd is going to take you through on Wednesday, going through how selection can shape the genetic variance covariance matrix. It's the first day back from Thanksgiving break. How do your brains feel after what we just did? Connor?
You know, I was really trying, but I definitely got lost a little bit.
Okay, all right. At the beginning of class on Wednesday, go through this material between now and Wednesday. There's also a homework assignment on Moodle. Start working on that. Work on that between now and Wednesday, and we can take this up on Wednesday, because I don't know that it's going to take us all of the 65 minutes to get through the adaptive landscape stuff. So we can answer questions about today's material on Wednesday. And also, when Dr. Judd goes through this on Wednesday, you might get a little better handle on this.
Yeah, I think that's a good idea. I can kind of tell the difference between how we've done this with a whiteboard versus over Zoom. It's a very different experience. So I think repetition will be a good way to do this. So go over that homework, watch this video, which I'll post this afternoon, and we'll get you there.
All right. I'll see my lab groups tomorrow at 7:45 on Zoom. How do I
had a question on is there the office hours still gonna be like the same, or because it's on Zoom?
Oh yeah, I meant to mention that. Clearly, we're not in our offices, so the way to do office hours is just make an appointment with us. So Dr. Judd's clearly in his house, I'm in my house. So we'll still be available during those times as well as outside of those times. It's just that we're probably not going to be hanging out on Zoom during office hours waiting for students to log in. So at least for the next 2 weeks, until the end of the finals, if you want to talk to us, you just need to make sure that we know you're coming.
Okay.
And we'll set up Zoom meetings for you whenever you need.
Okay. I've added a calendar link on the bottom of my email signature. So if you have a time that works for you, you can just plug it in there to my schedule directly and send me an email letting me know.
Okay. Thank you.
All right.